So start with the two that are furthest apart, right? So B and C are furthest apart. So I'm going to start with them. And I'm going to make those like my bookends, right? So I put those at the end. From there, it's kind of like solving a puzzle. You can use reference points. So A and C are 30 apart. So from here, I might divide this into thirds to help me just kind of identify where things are. 15, 30, like 15, 30, 45. Or sorry. Yeah, that works. 45. So A and C are 30 apart. So that means A is going to be over here, right? 30 from it. And then once I have A, I'm able to place others. Okay, so here's my A. I couldn't fit it all in one, or I had very small fonts. So here's A. And then it says that A and D are not 15. What were A and D? A and A and D are 25 apart. So then I can calculate, oh, I have it written there, sorry. So if this is at 15, 25 from there is D. And that satisfies also the 40. B and D were 40 apart. Okay. So start with your bookends, place them furthest apart, and then use those as reference points and go from A to B and A to C, D to A and D to C. So in the end, I have B, A, D, C. Usually, you're just asked to put them in the correct order. You're not actually, like on the AP test, they don't usually have you draw them out. Sometimes, but not like in the however many years I've been doing AP, I've only maybe seen them ask you to draw it once, but I've seen the order come up frequently. So even if your like positions are a little off, you have them in the same order. So how did they determine those frequencies of crossover in the first place? Oh no, this is a practice one. Sorry, I jumped out. Okay, so I want you to take a minute, pen and paper, and I want you to calculate the frequency, or not calculate frequencies. Jeez, Miss Smith. I want you to draw out this map. How do you think this map would look? What is the correct order? I'm going to give us 3 minutes. What is the correct order of the chromosomes? So Monsi, if you want to, oh, you might not be quite yet. Monsi, we are in the genetics agenda on page 10, slide 10, and we are making a map of the gene frequencies on that page. And I put 3 minutes on the clock. We're going to try to solve them together. Yes, sir, Alex. You've got it. So we got about 30 seconds. So if you started with your bookends, you place V and CN 45 apart. Okay, and then G was 15. So again, I decided to divide it into thirds to help me identify where things are. So G was 15 from V, and it was 30 from CN, correct? And then RB was 25, so between my 15 and 30. 25 from V was RB, and RB was also 20 from CN. So that satisfied that. And 1, 2, 3. What am I missing? 1, 2, 3, 4. 1 through 5. B, G, R, B, C, N. Okay. So the order would have been V. I didn't change my share while I was drawing. So the order would have been V, G, R, B, C, N. Correct, correct, correct. Good job. Okay, so point for you. That's a quiz. I should do that. Okay, so then now this one is actually calculating the recombination frequency. So that's maybe more difficult than the mapping. If we were in class and we had extra days, I give you a really difficult map one to do, but we're not going to do that because we don't need to. So how do we calculate that frequency of crossover? How often it occurs, right? So what we want to do is, of course, this is going to happen. You're going to look at a trait that is not just monohybrid, right? You wouldn't see a crossover if you didn't have 2 reference points. So you have to look at 2 different traits that are appearing or seem to appear on the same chromosome. And what makes them seem to appear on the same chromosome? Simply that they seem to always be inherited together. Like if you have blue hair or blonde hair, you almost always have blue eyes, right? Blonde hair, blue eyes. So it would be a fair guess that those 2 are inherited together, right? Because I see them appearing together all the time. So when you don't see a mix match, gene traits are probably linked. So you're going to take a dihybrid organism like purple flowers that are axial, and you're going to cross them with a recessive individual, right? That guarantees the recessive trait shows. That's kind of like your test cross, okay? So I'm going to start in this example because I stole it from the internet. It uses an alien. So I'm crossing a purple-haired, round-headed alien with a yellow-haired, flat-headed alien. So this individual is homozygous recessive, and the original parent is a heterozygous. So this guarantees I'm going to see those recessive traits show up. So if there's 1,000 offspring, here's what I noticed. Okay, so if these were linked, right, the purple hair should only show with round head, and the yellow hair should only show with flat heads. If they're linked, this P, this recessive P and this recessive R are stuck together, right? They don't separate. So I should only see the two versions. Purple haired round, yellow flat. But I don't. I see this mismatch in the middle. Okay, so we can assume the phenotype that is in the greatest percentage is the original form. The phenotype that is in the smaller percentage is the recombinant. Okay. And that's what this is showing here. This is talking about the fact that those genes should always be inherited together. And so you have an overrepresentation, meaning a greater percentage. And then the recombinants are going to be the lower percentages, far fewer. So what you're going to do, you're going to add up all of those that seem to have crossed. So the mismatched group. Divide that by the total number of the offspring. So you had 1,000 offspring. How many of them showed a mismatch? How many of them were not the way you expected them to be? How many of them had recombinations? Okay, so in the middle, you had the mismatch, round and yellow, flat, and purple. Okay, you had 100 of one form and 98 of the other. So there's 198 recombination events. Okay, we have 1,000 total, I believe, if this adds up to 1,000. So divide recombination by total individuals, and you get your frequency, 0.2. So this tells us, and frequencies, when you see that word, they're going to be decimal forms. They're not, like when you say percentage, you're multiplying by 100. You don't multiply the frequencies, you leave them in decimals. So this tells me that the color of the hair and the shape of the head are 2 or 0.2, right, centimorgans apart. So 20 centimorgans apart, because that's our frequency. And you multiply that by 100 to get your centimorgans. So 20 units apart. Okay. Any questions on that one? Okay, so maybe before I do the chi-square one, which screen am I on right now? Oh, good. Okay. Go to the Biozone gene mapping. Pull that up. This explains the chromosome maps. Okay, up top, this explains the crossover calculation. Here's an example. Here, I'm going to put you guys into groups, and I want you to finish. I want you to do the second page. I put it in as a Cami, so so you can talk those out a bit. Then I have to stop sharing. Okay, breakout rooms. I'm going to do 5 rooms. I'm going to have you choose if you can. So the rooms are open. And if we'll put Abigail in room 1. I forget, I kind of need to do this. Ahmed in room 2. Alex is in room 3. Choi is room 4. And Cameron is room 5. So you can, if you have the abilities to join a friend, join a friend. Otherwise, I'll go through and sort you out. So right now we're just doing the gene mapping page. Okay, so the way I solved it, it was nice because these maps actually had unit marks. And so you can maybe just check your work here. If I did it right, I didn't draw it out in advance, so I did it while you guys were working. So your map should look something like this.
For gene map too, does it matter where you put C? On like either side of, I think it's M.
Right. We don't know for sure since it doesn't have a second reference point. We're just taking a general rule that the furthest ones are the bookmarks.
Okay, thank you.
And I would think since there isn't a reference point on the other side, that probably means that it's closer to that one. Otherwise, there would probably be crossover. And the frequency is 0.17. Did you guys get that? Your quiz today is just chi-square. But this is something, you know, we're always preparing for May.
Okay.
So it's something you may see then. Okay. I think your quiz may or may not have the crossovers, but it's not like you're calculating them. You're just using them as one of your 4 variations. I forget. The numbers are given to you. You wouldn't have to calculate that. You're just calculating. It's just working with the chi-square formula. Okay. So do you guys want to do one of those? Looking at the time, maybe we don't want to do the second one as a group, which you guys didn't really interact with each other as far as I could tell. But do you want to do one here before I turn you loose? Probably, right? Okay. So again, the fruit fly, quite often we use the fruit fly. There's a whole lab we're supposed to do, but we just don't have time. So normal wing female with sepia eyes is crossed with a vestigial winged male, and their offspring all come out red-eyed and normal. So that tells me I'm working with homozygous individuals, right? If all of the F1 generation comes out the same. Yeah? Yeah, okay. So this is classical Mendelian, where you got your P generation, which are true breeds, and then your F1 generation, which is a heterozygous cross. Okay, so now we're going to go on to the F2 generation. Usually you have to look at the F2 generation in order to determine the inheritance pattern. And this would have been a good quiz question. But I told you guys, all you have to do is work with the formula. So I'm not having you go back further and try to determine inheritance pattern, but this is what you would want to do. So, so from a problem like this, you need to be able to, for the future, determine a mode of inheritance. So that's like saying, are they linked? Do they sort independently? If we're looking at 2 different traits. If it was just one trait, are they multiple alleles? Are they incomplete? Are they codominant? Are they complete dominance, right? So that's what you would look at for a single trait. This is a double trait, a dihybrid. So that means either they're sorting independently or they're linked. So your hypothesis has to be one of those 2. And remember, your null hypothesis states that there's no difference between your hypothesis and the expected outcome. So what do you think? Do you think this one is sorting independently, or do you think this one is linked? I'll open up my chat box if you want to secretly make a guess. Alex, your last name just really suits you. How many people think you can use your reactions if you want? How many people think that this is a linked trait, like what we just did? That last problem we did on the worksheet was a linked trait. How many people think this might be linked? Okay, I'm getting a couple of answers now in the chat box. Okay, give me a thumbs up or a raised hand in your reactions if you think this is sorting independently. So if you're not sure, you always want to do a Punnett square to see. So do a Punnett square, assuming it's linked, see if the traits match. Do a Punnett square independently assorting, and then see if the results match. So that's the best way to check your guess, is to do a Punnett square and see if the numbers agree. So when I did the Punnett square, I got, if they were linked, I got a 3/4 showed the dominant trait for both, and 1/4 showed the recessive trait for both. So does this look like a 75-25 split? No. So then I did the square, and for if I did a dihybrid cross with my 16 boxes, meaning they sort independently, I got a 9:3 to 3:1 ratio. Does this look like it might be a 9:3 to 3:1 ratio? Yeah, and the fact that I got all kinds of different variations, right? So in fact, if this was 9:3 to 3:1, I would expect 55, 18, 18, and 6, I think. So that's pretty close to those numbers that I was expecting. So to do chi-square then, I just created my expected results based on my box, right? A 9:3 to 3:1 ratio. If I calculated this correctly, there were 98. There should be 100. What did I do wrong? 58 and 20, 17. Because I wrote 3 instead of 5. Oh, good. So there's 100. My numbers might be changed. So there were 100 total. So I would expect 9/16 to be dominant, 3/16 to be dominant recessive, 3/16 to be recessive dominant, and 1/16 to be recessive. Okay. So can you real quick calculate out the chi-square? Tell me what chi-square would be for this problem if your hypothesis, your null hypothesis, is that the F2 generation would show a 9:3 to 3:1 ratio. So the statement would be, if these genes sort independently, then the F2 generation would show a 9:3 to 3:1 ratio. Here's your observed values. That's what you actually see. Your expected values are based on your hypothesis of a 9:3 to 3:1 ratio. There's 100 total, so it makes it pretty easy math. So I'm doing 9 divided by 16 to get my 9/16. Multiply that by 58 to get my expected number. No, sorry, multiply by 100. So in this case, we expect 56.25. I'm going to keep my decimals because if I drop my decimals too early, May, you doing the math? If I drop my decimals too early, I likely end up with the wrong answer in the end. If an expected value isn't given to you, you would have to do a Punnett square to determine your expected value. So can somebody put in the chat box what they got for the chi-square so I can see if my math seems like it might be on the right track? I decided I don't like doing math on my phone. That's what I learned from this. When you got a chi-square, throw it in the box, please. So I'm expecting a 9:3 to 3:1 ratio if they're sorting independently. Micah, don't round up. And then that's out of 16, right? So 9/16, 3/16, 3/16, 1/16. Multiply by 100 because there was 100 individuals. So how many out of the 100 did you expect to show that value? So if I did my math right, we have 56.25 that we expect to show the dominant traits. We have 18.75 that we would expect a dominant recessive and a recessive dominant. And the 1/16 would be 6.25 individuals that would show the homozygous recessive trait. So first of all, did you get the correct expected value? So then that's my expected value. My observed values were 58, 20, 17, and 5. I expected 56.25, 18.75, and 6.25. So then I did observed minus expected, square that first. That gets rid of your negative values if you have them. Divide that by the expected value. So remember, this one is your observed, this one is your expected. I believe I got 0.544. I always carry a couple extra decimals. 0.0833. 0.1633, 0.25, and when I added those up, I had 0.551, I think, but whoa.
So Miss Snook,
Yes.
on the so
I
like
don't know how to. So
Miss
if you got a different number, you want to find out. Yep, just one second. You want to find out where it is you're making your mistake. Did you make a mistake in the expected value calculation? Or did you make a mistake in calculating the individual probabilities? Or did you just not think to add them all up? Did you do them all at once? Did you do them separate? So you want to figure out where along the lines you make your mistake. Okay, Ahmed.
Yeah, for the first value, 58 and 56.25, if you do it, it comes out to be 0.0544, not 0.5.
Oh, I wrote it wrong. Okay, thank you. 0.0. Thank you. So 56.25 observed. Observed is 58 minus expected, 56.25 equals, and then I got to square it, which means I got to go to my scientific calculator. And I got 3 point whatever divided by observed minus expected squared divided by expected, 56.25. Yes, I missed writing a 0. Thank you. Any questions on how I got any of the numbers I got? Okay, so for the sake of time, our quiz was available 10 minutes ago. Because you're going to be in your locked version, right? You're going to want to, did I not have my screen? Oh, it's because I closed it. This is the AP Central Equation Sheet, formula sheet. You want to be very familiar with that and where you can find information. This is what they provide you in May. So you want to have the formula sheet nearby when you do your question because it's going to ask you for degree of freedom, chi-square, or critical values, right? So we need this piece of information. You can print it, or because you're in that lockdown browser, right, you wouldn't be able to have two at the same time. If you wanted to just take a picture of your chi-square table, you could do that. Because the questions, like using I didn't have a way of putting the formula in there for you. So you might want to just snap a shot of that chi-square. So we did linkage, we did frequency, we did figuring out a genetic inheritance pattern. And now you guys have a quiz to take. There's only 4 questions. It's short. And then over the weekend, the Sordaria lab. What questions do you have?